Hello, everyone. My name is Yingxin, and I'm currently a PhD student at UC Irvine. Today is a great pleasure to be here to share our recent project, SAILER, Scalable and Accurate Invariant Representation Learning for Single-Cell ATACseq Processing and Integration. Okay, I'll start with some background. So single-cell ATACseq measures the chromatin accessibility at single-cell resolution. If we have reads at certain regions, it means that this region is determined at open states. And proteins that regulate genetic processes usually bind to open chromatin regions. The chromatin state for each cell can be represented as a binary vector. So regions with signal denoted as 1, otherwise 0. And the entire dataset is thus n by m binary matrix with n cells and m peaks. The matrix is usually high dimensional. M and n can both go over 100,000. Also, it's sparse, so it's impossible to directly interpret the raw data. So usually the first step for data analysis is to perform dimensional reduction to get to a lower dimensional embedding. Usually this lower dimensional embedding is biologically more meaningful and less noisy. With a good embedding, we can then run clustering to annotate cell types. This cell type information could then facilitate researchers to fully utilize the power of single cells for many downstream analysis, like differentially accessible regions between different types of cells to identify regulatory elements specific for each cell type, or inferencing cells' developmental trajectories to analyze continuous changes of chromatin accessibility within cell population. However, all these analyses require a good lower dimensional embedding to begin with. So why learning a good embedding is so hard? We think partially it's due to the technical variations introduced at the sequencing stage. Here we show some statistics of two single-cell ATACseq data from the same tissue. So they are both mouse brain, but sequenced with different platforms. On the left column, you can see the read depths as UMI in x-axis plotted against promoter ratio. So each point represents a cell. On the right column, we show the statistics, the bar plots of UMI and promoter ratio of two cross-platform datasets. From the figure, we can first see a clear batch effect. So read depths from different sequencing platforms are of different magnitude, and the promoter ratios are also different. Also, we can see great read depth variation even within the same sample. Read depths could change dramatically. And last but not least, for single-cell ATACseq, we also have the stochastic dropout issue. That requires imputation before any downstream analysis. Here we show how these variations affect the dimensional reduction. The first example is about batch effect. Here we show the t-SNE visualization of latent embedding learned by a deep gentry model without batch effect correction. This is on the cross-platform datasets we just mentioned. It's from the same tissue but sequenced with different platforms. Without specific mechanism dealing with batch-related confounding factors, when jointly analyzing these two datasets, clusters are predominantly driven by simple batch effects rather than the true biological states of cells. The second example is about read depth variation. We've seen that if we don't correct for batch effect, we will get problematic embedding result. Here, even within the same sample, read depth variation can be a confounding factor affecting the dimensional reduction. Here we show the UMAP visualization of embeddings generated by a simple linear method, the latent semantic indexing on two simulation datasets. The left one is simulated without depth variation. So all the cells have same number of fragments. The right one is simulated with depth variation. The read depth of each cell in the simulation is sampled from a log normal distribution. When there's no depth variation, LSI could distinguish the real cell type quite well, which takes totally 1% of the total population. However, when there's depth variation, the real cell type cannot be found. So with this in mind, we are trying to develop a computational method that could tackle these technical effects, such as batch effect and redepth variation. In the meantime, scales well with large datasets. Our method is based on a variant of variational autoencoder. Variational autoencoder consists of an inference model and a generative model. The inference model learns a posterior distribution of latent variable Z parameterized by a neural network. The generative model samples from the latent distribution through reparameterization and then reconstruct X through a neural net. The latent variable Z is usually of lower dimension, serving as a bottleneck. It's usually used for dimensional reduction in unsupervised settings. According to the goal of dimensional reduction, we want the latent representation to be informative on cell state or cell type. However, in real world, besides the cell identity, other technical variations are also involved in the data generation process, such as redepth variations or batch effect, etc. And these technical variations can be observed when we collect the data and are not related to intrinsic biology we are interested in. However, if we do nothing about it, it will be coupled with the latent representation and affect the final result for dimensional reduction. We call these technical variations as confounding factors. So here comes our method. We want to decouple these confounding variables with a true biological state during our training. And we achieve this through the idea of invariant representation learning. The goal is for the inference model to learn a latent representation Z, which is independent with the confounding factors. So intuitively, we want the invariant representation Z reflects the intrinsic cell states and does not affect by the technical variations, such as sequencing depth and batch effect. We achieve this by adding an extra penalty on mutual information between the invariant representation Z and the confounding variable C. So here comes our overall framework. SAILER could take data from multiple sources as input. Redepth of each cell is converted to normalized log library size, and batch information is denoted as binary encoding. The objective function is negative log likelihood with conditional reconstruction plus the mutual information penalty between the latent representation Z and the confounding variable C. During the training, SAILER simultaneously deconfound depth variations and batch effect in the input data and learns the latent representation Z invariant to changes of these confounding factors. We first experimented SAILER on simulated datasets. We benchmarked against a simple linear method, LSI, and also scale method, which is previously proposed a variational autoencoder-based method on single-cell ATACseq. We use 3 simulated datasets and visualize embeddings with UMAP. The first dataset in example A contains 1 real cell type along with 4 major cell types. Deep learning methods can capture the real cell type pretty well, while the linear method, LSI, couldn't distinguish it. When we look at the redepths visualized by color map on the embedding, we can see LSI and scale does not mix redepths well. So we can observe elongated cluster shapes and lower to higher redepth distribution within each cluster. This could impact the clustering performance and also impact the imputation process since the neighboring cells in the embedding all have similar redepths. However, in SAILER, with invariant representation learning, SAILER mixes data well for different redepths and generates better embeddings. This is also in line with the quantitative measurements shown in Table 1. where SAILER has the lowest mean mutual information between latent representations and the read depths. For the second dataset, we simulated a harder case with more cell types and shallower sequencing depths. In example B, we can observe similar trend in the results. In the third simulation, we simulated a dataset with 2 batches. 2 batches have different average read depths and different signal-to-noise ratio. Also, each batch has one unique real cell type. As we can see in the results, SAILER preserves the real cell type better for both datasets and makes its data well for cell coming from different batches. This is also supported by the lowest mean mutual information between the latent embedding and the one-hot batch information. For methods that are not dealing with batch effect, we can see data from different batches forming their own clusters, which is not favorable when annotating cell types for downstream analysis. We then experimented SAILER on real datasets. We first tested on the mouse atlas dataset, which contains over 80,000 cells and 40 cell types. This dataset has been previously used for benchmarking 16 different single-cell ATACseq computational methods. SNAP ATAC is one of the leading methods, and also the only method could finish processing this entire dataset in around 11 hours. In terms of clustering metrics, SAILER achieves better quantitative scores at ARI and NMI. And from the figure, we can see SAILER generating more separable clusters compared with SNAP ATAC and Scale. Also, SAILER has lower mean mutual information between latent embedding and read depths. This corresponds to the better mixing in read depth distribution within each cell cluster. We also use SAILER to merge 2 mouse brain datasets. Compared with Scale method, SAILER with invariant representation learning merges clusters from different batches together. We then run clustering on our embedding and visualize the gene activity scores of some marker genes to validate our clustering results. And it appears our method generates reasonable clustering results after integrating 2 datasets together. Well, without batch effect correction, Scale's cluster annotation is impacted by batch effect. Moreover, SAILER could be used for imputing single-cell ATACseq data which suffer from sparsity due to dropout during the sequencing. Since in SAILER, we have control over these confounding variables at imputation stage, we can reconstruct the data free of various confounding factors. We tested on the simulated data. The raw simulated data is sampled from foreground peaks of bulk ATACseq data and then combined with uniform noise. The simulated data contains 2 batches simulated with different depths and signal-to-noise ratio. After training, we then run imputation on the raw data with Scale, Magic, and SAILER respectively, and reduce the dimension of the imputed data with PCA as well as the raw data. We see that the data imputed by SAILER are not affected by batch effect and preserve the real cell type quite well. Without correcting for read depths and batch effect, methods like Magic and Scale tend to impute data based on their neighbors in the embedding with similar read depths and batch signature, which is not favorable for downstream analysis. And since the simulated data is generated from the bulk data, we know the ground truth, so we can compute the die-score between imputed single-cell data and corresponding type of bulk data. So die-score shows how well imputed data overlap with ground truth. It's a well-balanced metric, and we can see SAILER helps with dropout issue if we compare its die-score against raw data. Also, SAILER outperforms Scales in terms of the die-score. An advantage of deep learning-based method is mini-batch training, so it doesn't involve entire matrix factorization. So usually runtime for deep learning-based methods scales linearly with sample size. As you can see, epoch time of SAILER scales linearly up to 1 million cells in our experiments. And some traditional methods like SNAP ATAC doesn't have these properties. So deep learning methods like SAILER are more efficient when analyzing large-scale datasets. For example, on Atlas level datasets or integrating multiple samples. And that's all contents for today. Before I end, I'd like to thank the co-author Lai Yi for this paper, and also thank my advisors, Professor Xiaohui Shi and Professor Qing Nie, and our collaborator, Professor Jin Zhang, on this project. Also thank folks from the lab for all the comments and suggestions. And that's all for today's presentation, and thank you all for watching.